So what better way to learn more about them than through photos, right? So this first lab activity will be the starting point for all the lab exercises that follow, because it focused on the external morphology of insects and how each physical part contributes to their function, lifestyle, and their survival. So you had to refer to a video by the SciBugs channel as well as a separate material with all those basic morphologies drawn in cartoon form. So that includes the antennae, mouth parts, ovipositor, wings, legs, and eyes. Okay, then we mined the internet for various images of those insects containing the morphologies that were zoomed in so that we could refer to it and then recreate it with pen and pencil, then color each part differently to distinguish them apart, of course. And then we also had to include their core functions, such as if that part is for sensing, locomotion, protection, and the likes. So all in all, this activity tells us not to be fooled by the size. Definitely, no matter how minute that part can be, it can actually be an intricate structure that helps the insect adapt to an ever volatile environment.
Our laboratory exercise 3 was focused on the basic collection of arthropods, particularly in insects. The main goal of this activity was to familiarize us with various collection methods and to understand the diversity of arthropods within our surroundings. We conducted 2 types of sampling. First is the active sampling, which involves direct methods like hand collection and sweep netting, and passive sampling, which uses traps such as baited, pitted traps. These approaches help us to observe how different species respond to various collection techniques. This activity is important because collecting and identifying arthropods provides valuable insights into ecosystem balance, biodiversity, and even pest control management. It also trains us to apply proper scientific technique in fieldwork and ensuring accuracy and safety specimen collection of these species.
In order to collect various arthropods, certain equipment is necessary in order to bait and lure them. There are many methods of collecting arthropods, but the focus here will be manual collection, sweep method, and bait method. For the manual collection, forceps are used to grasp any specimens found, coupled with vials to store them in. A fine paintbrush can also be used to pick up any of the specimens. For the sweep method, a large insect net is used to physically collect specimens by sweeping the air or against any shrubbery. The baiting method uses a mixture of tuna and peanut butter to create a bait, where it will emit a pungent odor that will attract any specimens. For the temporary storage of all these specimens, a Ziploc bag can be used. Alcohol is used to clean a specimen or it can be used as a killing agent, but most importantly, it acts as a preservative. Once the desired specimens have been collected, any live ones will be transferred to a killing jar, which consists of cardboard on top of cotton balls that are soaked with a fumigant, meant to immobilize and suffocate the specimens and minimize any damage done to their bodies. For preservation, the specimens can be stored in vials, where the specimens is submerged in 70% alcohol.
Laboratory 4B focuses on the systematic identification of arthropods. The step-by-step process begins with detailed observation of key distinguishing characteristics like body segmentation and mouth parts to accurately place the specimens into the correct order and family. This information is then used to construct its position on the taxonomy tree, moving precisely down to the genus and species. Now, an incorrect classification can lead to a critical failure in controlling vectors. Therefore, our skills in precise taxonomic identification is a direct input for successful public health intervention and disease control.
So in insect specimen mounting, you will be needing several materials. You will need an insect specimen. This is what you will be viewing. You will also need paper clips made by entopunchers. This is where you will be adhering your insect specimen using this water-based glue. Next, you will be needing entomological pins to pin the paper tip using a pinning block. A pinning block helps you set the insects and their labels at uniform and correct heights in a pin. Next are your entomological probes. They are used to manipulate your insects to have the better orientation when viewing under the dissecting microscope. A Gucci ball is where you will be able to freely manipulate the orientation of your insect when you are viewing them under the microscope.
For Laboratory 4D, we explore the key morphological characters of arthropods used for identification. Here's a quick rundown on their general and surface structures. Insects have three main body regions or stigmata, which are the head, the thorax, and the abdomen. The head holds sensory organs and mouthparts, while the thorax is divided into the prothorax, mesothorax, and metathorax. It bears the legs and the wings. The winged insects are called Adaleine, wingless ones are Acarus, and Daolate are insects that have shed their wings. Their top exoskeleton is made up of hardened sclerites joined by sutures with a sternite below, plurite on the sides, and turdite on top. Surface structures differ. Some are rugose, foveate, or granulopunctate, while others are glabrous, pubescent, or pilositive. These external features serve as important identification tools that highlight the diversity and adaptability of arthropods.
Let's take a quick look at the arthropod head, a powerhouse of sensory and feeling structures. The head holds the key parts like the antennae attached through the scape, funiculus, and flagellum, all fitting into grooves called the antennal fossa or scroll. The eyes with many facets called ommatidia and the single ocelli help detect light and movement. Moving down, the frons, snipus, and labrum frame the mouth parts, which include mandibles, maxillae, and labial, each with sensory bulbs that guide feeding. Structures like carina, vortex, and occiput define the head shape, while internal lines like the apophyseal lines and tentorial bit guide support. In short, the arthropod head is a compact command center built for sensing, eating, and surviving.
Moving on to the insect thorax, it is the center of movement and locomotion, composed of three segments. We have prothorax, mesothorax, and metathorax, each bearing a pair of legs, and the last two often carrying wings. Key structure including the coxa, trochanter, femur, tibia, tarsus, and tarsoplas, which make up the leg segments for walking and gripping surfaces. The spiracles allow respiration, while the glands such as the metapleural and metatibial glands secrete protective substances. Sclerites like the notum, epimeron, and the catepisternum form the exoskeletal plates, giving rigidity and muscle attachment style. Internal grooves and sulci, such as the metanotal groove, help with flexibility during movement. Overall, the thorax provides strength, coordination, and power of the insect's essential activities of walking, flying, and survival.
In entomology, the abdomen is the last major body segment of an insect, found behind the thorax. It houses vital organs for digestion, excretion, and reproduction, and includes key structures like sting, ovipositor, and breathing openings called spiracles. Its flexibility allows insects to move and perform important functions such as mating and defense. Overall, the abdomen plays a crucial role in maintaining life and survival, making it one of the most essential parts of an insect's body.